A very warm welcome, dear listeners. I am Piyush Priya, a researcher working in the area of bioinformatics and computational biology at National Institute of Plant Genome Research, New Delhi, India. In this video, I would like to give you all a basic introduction about combined stresses in plant and how the emerging field of bioinformatics can play an important role in its research. So let's first see what are the major threats faced by plant in nature. Plants are sessile, which means they cannot move like us when faced with danger or alarms from the environment. Thus, in their natural environment, they experience many stresses. A stress can be defined as any external factor which is detrimental for the plant health. Majorly, these stresses are classified into two broad categories. The first being the abiotic category, when these stress originate from the surrounding environment, for example, chilling, cold, heat, flood, salinity, heavy metal, highlight, and rot. When this stress originate from a living organism like bacteria, virus, fungi, and nematode, they are called as the biotic stresses. Together, these individual stress cause severe damage to plant yield and productivity. Dear listeners, in nature, the occurrence of these abiotic and biotic stresses in isolation is rare, which means that plants are often challenged by these stresses in combination. This is what we call as combined stresses in plants. These combination of stresses can be of abiotic stress combination type. For example, heat and drought stress coming together to plant. Secondly, it can be of biotic stress combination type. For example, the bacterial and fungal stress coming together to plant. And finally, it can be of abiotic and biotic stress combination type. For example, drought and viral stress coming together to plant. Often, these combined stresses are more detrimental to plant yield and productivity and hence needs dedicated research. Before we find solutions, it is important to understand the problems. So next we will see what are the global challenges that a plant biologist actually face. The first being the climate change. It is projected that by 2050, the average global temperature will rise to 16 degrees centigrade. The next challenge is of pest. An estimate suggests that by 2050, the amount of global crop production lost to pest will increase to 20 to 25%. By 2050, the rise of global water demand will also increase. The rise of global population by 2050 is estimated to 8.8 billion, and a challenge is to feed such a huge population. And then by 2050, there will be a reduction in the availability of the arable land. So it is important for the plant biologists to find solutions to these problems. To address these challenges, plant breeders are developing improved plant varieties which can withstand pests and diseases with fewer crop inputs like water and pesticides. Researchers also are developing resource-efficient crops and plants that are adaptable to the changing climate conditions. Next, we will see how in this pursuit the emerging field of bioinformatics can play a vital role. But before that, let's have a brief introduction about bioinformatics. So, bioinformatics, as the term suggests, is made up of two words. The first being the biology, which we all know is the study of the life, and second being the informatics, a term used for computer science and information technology. So when we apply computational power to better understand the biological data, it is called as bioinformatics. Next, it is important for us to understand what kind of biological data we study in bioinformatics. To understand this, let's first see the basic architecture of a plant cell. We all know that a plant cell is encapsulated within a cell wall and cell membrane and has other important organelles like blasts, vacuole, and nucleus. It is in these nucleus where the DNA intertwined in the form of chromosome present. The DNA act as a blueprint for any living organism like plant. The term DNA stands for the deoxyribonucleic acid, a double helical spiral made up of four letters ATGC. So next we will see why bioinformatics is so important in today's research. But before that, let's know some more interesting facts about DNA. Your DNA is like an instruction book. It contains instruction about how any organism will develop, grow, and function. Variation in these instructions leads to variation among us. And complete set of these DNA instruction for a living organism is called as its genome. If we want to understand about how a particular living organism like a plant function, we need to study its full complement of DNA instruction called genome. It is from here that the concept of the genome sequencing originated. Just as your eye scans a sequence of letters to read a sentence from a book, the sequencing machines read a sequence of DNA bases, that is ATGC. Reading through the genome of the organism is called whole genome sequencing. Initially, however, it was a very tedious task and took really long time to sequence any particular genome. But with recent technological advancement, it is just a matter of few hours. This has ultimately led to enormous rise in the genomic biological data. By sequencing a particular genome, scientists try to find important functional units like genes, how genes work together to direct growth and development, maintenance of an entire organism, and also study non-coding regions present within any genome. The current challenge is to store, transfer, and analyze and display these genomic data. This is what necessitated the use of the bioinformatics, in which scientists use the computational power to better organize, catalog, and decipher the big biological data. We have been talking about the biological data, but let's have a look what it actually is. To understand it, let's first have a brief overview of the central dogma of life, which most of you might have surely heard about. It means that the information passes from DNA to protein via RNA. Hence, the dogma is a framework for us to understand the transfer of sequence information. Here in this workflow, we see the term omics used widely. Initially, the term omics was used to signify lots and lots of money. In the biological context, the lots of genes makes the genome of the organism. Gene transfer their information to the mRNA by the process called transcription. Again, lots of mRNA make up the transcriptome of the particular organism. When the mRNA passes on the hereditary information to the protein by the process called translation, and the whole complement of the protein is called proteome. The proteins by cellular processing form the metabolite, which together are called as the metabolome. And finally, the external manifestation, or simply how a particular organism will look and behave in its environment, is called as its phenotype. The present slide highlights some of the important role bioinformatics can play in plant biology research. The first role is in the development of databases, which is a repository to store, organize, and analyze the large amount of biological data. Few of the commonly used databases include NCBI, Phytosome Database, TIA Database, and the PDB database. The second important role of bioinformatics is to provide tools that will help in the analysis of biological data and bring out meaningful information from them. Some of the important tools widely used are BLAST and CLUSTAL-W. The third important role of bioinformatics is in the area of genome sequencing. Bioinformatics expertise is widely used to sequence model crops like Arabidopsis and rice. This aids researchers in finding valuable traits for crop improvement. Next, the knowledge gained from the model crops are passed on to the other new crops by a process called as comparative genomics, which aids in their improvement. Few examples includes development of insect-resistant crops like BT cotton, improvement of nutritional quality, development of crops that are able to utilize the resources in a much better way. To summarize this video, the slide here presents few important points. The biological science is under rapid transformation due to the technological advancement, and we are in a data-driven era. The emerging field of the bioinformatics is about using computational techniques to solve biological problems and represents the future of the biology. The Human Genome Project has fundamentally changed the biological science. To fully understand the cellular behavior, we need to elucidate information encoded in the genome and understand working molecules encoded by the genome, which calls for the process of the genome sequencing. Over the years, many genomes for the model crops have been thoroughly studied, for example, of Arabidopsis and rice. For a new protein, scientists can possibly predict its function by identifying well-studied proteins in other organisms that have high sequence similarity by the process called as comparative genomics. Towards end, let's know some of the interesting facts related to bioinformatics. The major push towards the rapid development of the field of the bioinformatics was the Human Genome Project that was completed in the year 2013. Our genome is made up of over 3 billions of these genetic letters that are ATGC. We have typically about 10 trillion cells, and if we could stretch the DNA from all these cells together, we can measure the distance between the Earth and Sun a whopping 100 times. Doesn't that sound interesting? By 2050, the 10 to the power 18 exabytes of genomic data will be produced worldwide. The question arises, how do we efficiently store it? The genomic data produced roughly doubles every year and requires novel solution from bioinformatics for efficient storage. Do you know that the first free living organism to be sequenced is the bacterium Haemophilus influenzae? Similarly, the first eukaryotic genome sequenced was of Saccharomyces cerevisiae. The first multicellular eukaryote to be sequenced was of C. elegans, and the first plant genome to be completely sequenced was of Arabidopsis thaliana. Margaret Dayoff is known as the mother and the father of the bioinformatics for her work in which she completed the atlas of protein sequence and structure in her book. Dear listeners, I would also like to suggest you certain web resources which are having exciting and interesting bioinformatics activities for the school kids. This will definitely give you a flavor of the basic bioinformatics analysis that are usually performed by any bioinformatician. At last, I would sincerely acknowledge the funders of this project, the National Institute of Plant Genome Research, New Delhi, India, and Department of Biotechnology, Ministry of Science and Technology. Thank you for your patience, and I would like to end by the famous quote of the Albert Einstein. The important thing is to never stop questioning. This is what makes and drives the science to prevail.